Very good afternoon to one and all present here. Myself, Dr. Dilesh Singhvi. I extend a warm welcome to all in APTI sponsored online national conference organized by Department of Pharmaceutics and IQAC of Vidarbh Youth Welfare Societies, Institute of Pharmaceutical Education and Research, Gurgaon Meghe Wardha, on nanotechnology in healthcare. I find immense pleasure in introducing this session speaker, Dr. Sayog Jain sir, Professor Naipar Mohali, who is going to tell us about design and development of nanotechnology-based drug delivery strategies for advanced healthcare. Dr. Sayog Jain sir is working as a professor at Center for Pharmaceutical Technology, Department of Pharmaceutics of National Institute of Pharmaceutical Education and Research, Mohali, Punjab, India. Previously, he worked as scientist at Defense Research and Development Organization, Ministry of Defense, Government of India. His key research interests include exploring different types of nanodrug delivery system for various medical applications, including targeted anti-cancer drug delivery, oral delivery of vaccines, peptides, and therapeutics, co-encapsulation of high-dose drugs, especially antioxidants, topical and transdermal drug delivery, gene delivery, and carbon nanotubes. He has patented several nanotechnology-based drug delivery platforms and licensed tamoxifen SES technology for improved breast cancer therapy to industry for commercialization. Dr. Jain is the inventor of 14 granted patents. He has authored 2 books, 11 book chapters, and published over 230 research papers in high-impact international journals, H-Index 62, with more than 12,700 citations. Dr. Jain has won several prestigious awards, including AIPS Distinguished Young Educator and Research Scholarship Award, ABAB Gold Medal for Advanced Research in Nanoscience and Technology, Shivnath Rai Kohli Memorial Mid-Career Best Scientist Award, Pharma Innova Award for Best Research Guide 3 times in 2014-15, 16-17, and 17-18. Bharat Jyoti, the Glory of India Award, Rising Suns in Asia Awards, Alkyl Amine Young Scientist Award, INSA Medal for Young Scientist, OP Young Scientist Award, and Punjab State Young Scientist Award for his outstanding research in the field of nanomedicine and novel drug delivery systems. He also secured the rank in list of world's top 2% scientists in the field of pharmacy and pharmacology, published by Stanford University and elsewhere in 2020 and 21. Last year, he has received APS Fellow Award 2022, which was presented to him during annual APS conference held at Boston in October 2022. He has supervised 2 postdoc, 17 PhD, and 125 master level projects, and currently supervising 1 postdoc, 11 PhD, and 10 master projects. Dr. Jain is a widely traveled scientist, delivered over 170 invited talks across the globe at numerous prestigious platforms. His research groups has also been well funded, more than 13 crore Indian rupees by various government agencies and pharmaceutical industry. Dr. Jain is serving as expert committee member of Indian Pharmacopoeia and Radiopharmaceutical Committee of Department of Atomic Energy, DAE, the Government of India. He is editorial advisory board member of few international journals like Molecular Pharmaceutics, APS, PharmSci, Tech, then Drug Delivery and Translation Research, Biomed Research International. Before we start with the talk, let me briefly explain the rules to be followed during scientific session. Please keep your mic mute. Please be attentive. A feedback link will be posted in the chat box at the end of session, which contains specific question to the session. This link will be active for 30 minutes. Your attendance is compulsory for being eligible for certificate. Do not interrupt to talk by asking a question. If you have any question or comment for the speaker, please wait for the question and answer session. Now I request Dr. Sayyog Jain sir to deliver his valuable talk. Please, sir.
Sir, I think you are mute.
Sir, please unmute yourself. Sir, please unmute yourself.
Yeah. Thank you. So am I audible now?
Yes, sir.
Okay, thank you. So let me share my screen. Hello.
Sir, you are audible and screen is visible.
Okay,
Please,
thank you. So let me
click on
start.
a slideshow.
So,
Slideshow.
yeah. Okay, so thank you, Dr. Singhvi, for very nice introduction, and thank you for the kind of invitation to share some of my experience in the field of nanomedicine. Okay, I can see a lot of participants, more than 100 participants are there, and that is after lunch. So that is really impressive and commendable. Okay, so I will start with some basic introduction. This chart shows various challenges in the site-specific drug delivery. Okay, so most of the time we take drug to treat some infection. Okay, but in the conventional form, okay, most of the drugs they follow. Okay, they show the poor pharmacokinetic and unacceptable biodistribution and ultimately poor delivery at the site of action. So when we take the drug from the conventional formulation, so it reaches in the systemic circulation in the blood, and from blood it is rapidly eliminated from the body, okay, either by excretion or by metabolic degradation. And along with the blood, drug is distributed to various normal tissues, okay, non-infective tissue, so where it causes side effects or toxic reaction. Okay, and because of the several permeability related barrier, a very small amount of the drug is able to reach at the target site. Okay, so to deliver the drug, okay, in a minimum effective concentration at the target site, okay, we need to increase the dose of the drug. So if we increase the dose of the drug, then again the probability of drug reaching at the non-target site, it would increase, and the possibility or probability of the side effects will also increase. Okay, so can you imagine how much amount of that drug do we really need to treat any infection or to any disease? Okay, I can explain a very simple example is there. Okay, so there are total 10 to the power 13 cells in our body. Okay, so suppose if a person is suffering from some throat infection like common cold and cough, okay, in which few microbial cells they colonize in the throat region. Okay, so to treat that infection we take antibiotic. Okay, any antibiotic you can name. Okay, so suppose if we assume that one drug molecule will kill one cell, okay, then we will require total 10 to the power 13 drug molecules to kill all the cells in our body. Okay, so if we convert this number into the mole quantity of the drug, okay, we need to divide by this Avogadro's number. Okay, so this comes around 10 to the power minus 10 mole. Okay, if we want to convert this mole quantity into the gram or milligram quantity, we need to multiply by molecular weight of the drug. Okay, so molecular weight of most of the commonly used drug, it is in the range of 400 to 500 or maximum 600. But for simplicity of the calculation, let's assume the molecular weight of the drug is 1000. Okay, 1000 delta. Okay, then still this quantity comes around 16 nanogram. So theoretically, this 16 nanogram quantity is sufficient to kill all the cells in our body. Okay, but we take a huge quantity or huge dose of the drug. Okay, for antibiotic, as I said, okay, antibiotics are taken to treat the throat infection in which very few microbial cells they colonize in the throat region. So what is the dose of those antibiotics? Okay, minimum 300 mg tablet is available, 300 mg or 500 mg tablet is available. We take one tablet or two tablets per day for 3 to 5 days or 7 days. Okay, so theoretically we are taking a 3-gram quantity of the drug, approximately minimum 3-gram quantity of the drug. Okay, but theoretically this 16 nanogram quantity is sufficient to kill all the cells. So what is the rationale? Why we are taking this enormous dose? Okay, why the conventional dose or this theoretical dose it is not effective? Okay, so this is the problem. Okay, so this can also be explained by this cartoon. Okay, like there is enough food is available in the nest and with the mother, but kids are still hungry and crying for the food. Similarly, there is enough drug is available in our body, systemic body pool, but needy cell, okay, needy cell or target cell, they are not able to take up that drug. Okay, so that is the problem. We are not able to deliver the drug at the target site. So drug is roaming here and there in the body, and it is eliminating from the body, and it is unnecessarily causing the side effects and toxic reaction. Okay, so more than 99% of the drug gets waste and it fails to reach at the target site. Okay, another sad story of the conventional formulation is like side effects and toxicity. So it many time, okay, we need to discontinue with the therapy because side effects are so severe. Okay, so what can be the possible solution? Okay, one solution, very simple solution. Okay, we can synthesize the new drug molecule. That is new drug discovery approach. Okay, but it is not a very simple job. Okay, it requires almost 2 decades or 20 years of the extensive research, because starting from drug discovery, then preclinical testing, then phase 1, phase 2, and phase 3 clinical trial, then finally it comes to the FDA review and approval. Okay, so many steps or phases are involved. Okay, and out of the 2000 to 5000 lead molecule which are discovered in the early phase, hardly 1 or 2 molecules they are able to reach in the market. Okay, so by that time the molecule will lose its therapeutic importance. Okay, we all know there is no sure shot drug is available for HIV treatment. Okay, the reason is very simple. The HIV virus changes its configuration very rapidly. Okay, like recently, it is not particular to the HIV virus. All the virus, they changes their configuration very rapidly. Like recently, okay, a few years back or recently, we have seen that COVID virus. Every time within 2-3 months, the new variants, okay, used to come, and then speculations were there that this drug would be effective, this vaccine would be effective, this drug will not be effective. Okay, so this same applies to the HIV virus. So suppose if we develop or discover a particular drug based on the present structure or mutant of the virus, okay, after 20 years, how many times the virus would have been mutated, and we cannot expect that the drug would be effective at that time. Okay, this is the problem. Another sad story of this conventional formulation is this new drug discovery is the cost of developing the new drug molecule. So cost of new drug development, it is increasing day by day. Okay, so in the next coming year, the cost will cross the roof. Okay, this is the reason ki R&D companies, they are not able to afford the new drug discovery cost, so they are merging or they are being sold by some other big company. So this is the problem. So at this juncture, I would like to state, in the old Darwinian economics, the big eat the small, but in future, the fast will eat the slow. So for anyone to succeed, we need to be smarter and faster. Okay, so here I would like to present the concept of drug delivery. Okay, so instead of synthesizing the new drug molecule, we can improve the delivery of the existing molecule only with the help of some delivery system. Okay, so this concept is known as presentation of the old drug in new class. Okay, we are not making the new drug, but we are improving the delivery of the existing drug only by the formulation switch or formulation change. Okay, so this hypothesis is also known as magic bullet. Okay, German scientist Paul Aldrich gave this hypothesis, a magic bullet for the targeted drug delivery. Okay, so the drug is formulated in such a way that it will, when we inject inside the body, it will selectively go towards the intended site of action. Okay, it will not go into the other normal tissues or cells, so the side effects or toxicity can be overcome. Okay, so this is the extension of the first slide. This is very classical figure you might have seen in so many textbook as well. Okay, so when free drug is administered, it has poor affinity towards the target site. It has a greater affinity towards the non-target site, and it is rapidly inactivated or excreted or eliminated from the body. Okay, so the ultimate result is limited therapeutic effect and high toxicity. Okay, but when the same drug is put in some carrier system, okay, it is protected from elimination or premature degradation. It also limits the uptake by the non-target site, but it facilitates the transport of the drug towards the target site. So the ultimate result is a high therapeutic effect and a limited toxicity. So here this carrier system plays very important role in modulating the biodistribution or to achieving the targeted drug delivery. So these are nothing but these are the nanocarrier system. Okay, different type of nanoparticulate carrier system. Okay, they can be classified in different ways, but for simplicity, I have classified in 3 categories like vesicles. So that includes liposome and all other kinds of somes. Okay, these are lipid-based vesicle. Particle, so it includes a nanoparticle, carbon nanotube. Okay, nanoparticle can be made of polymer, they can be made of lipid as well, and the miscellaneous system, so that includes dendrimers, nanoemulsions, micelles, etc. So many new normal or novel carrier system they are being explored. Okay, but again, if we think we have prepared the drug loaded nanoparticle or liposome, okay, and we will be able to achieve the targeted drug delivery upon injection, okay, then it is absolutely no. Okay, we need to consider what would be the biological fate, okay, where this nanoparticle will go inside the body, how they will behave. Okay, so that we need to consider. Okay, so what happens when we inject this nanoparticle into the blood vessel, okay, intravenously? So this normal blood vessel, okay, they are composed of these endothelial cells, okay, which are very tightly arranged. Okay, so it is a very tight vasculature is there in normal condition. Okay, so this nanoparticle cannot go into the any other extravascular tissues. Okay, they will confine into the bloodstream only. Okay, however, the blood vessels supplying to this RES tissues, RES stands for reticuloendothelial system, okay, which comprise of liver and spleen mostly. Okay, so liver and spleen, okay, they have this fenestrated capillaries, means the endothelial cells, these are not tightly arranged, so these are having kind of leaky vasculature. Okay, so this nanoparticle, okay, will go into the liver and spleen. Okay, similar physiology is available in the tumor. Tumor also has this leaky vasculature because of the angiogenesis. Okay, so the blood vessels are not fully matured, so they are leaky in nature. Okay, this is the well-known fact. Okay, so what will happen? Okay, what would be the probability? Okay, whether this nanoparticle will go into the liver and spleen or RES tissue, or they will go into the tumor. Okay, so again, this is governed by this, like this blinking protein you can see. Okay, this is blinking. So these are called opsonin protein. Okay, so this opsonin protein, they recognize this nanoparticle as foreign particle. So when we inject this nanoparticle inside the blood, this opsonin protein immediately attack onto the nanoparticle. Okay, and they forcefully take them to the liver and spleen. Okay, so again, the probability of nanoparticle entering into the like a tumor vasculature, it is reduced. Okay, and after reaching into the liver and spleen, okay, liver and spleen, these are the two organs which are rich in the macrophage. Okay, so these macrophages, they internalize or phagocytize the nanoparticle by endocytosis process. Okay, and they degrade the nanoparticle. So this is the overall fate. Okay, so these nanoparticles are simply degraded. They are taken up by the macrophages in the liver of the liver and spleen, and they are degraded. Okay, so these nanoparticles, they are internalized by these macrophages by the process of known as endocytosis. It is called as receptor mediated endocytosis. Okay, so after internalization, it will lead to formation of the endosome. Endosome is then fused with the lysosome. Lysosome is a cellular machinery which is equipped with variety of enzyme which is capable of degrading this nanoparticle, okay, or any material. So these degraded nanoparticles are initially released in the cell cytoplasm, and then they are thrown outside the cell by known as exocytosis. I will show this one movie clip.
Animal cells to capture specific macromolecules.
Okay, so can you hear the computer voice?
For example, most mammalian cells use this
Here,
method
here,
to take up
getting that.
protein particles.
Okay,
The cell
you can see
uses
this movie clip, which is
receptors that specifically
self-explanatory.
recognize
How this
and bind to
endocytosis
the
takes place.
LDL particle. The receptors are clustered together in a reinforced membrane structure called a coated pit. An LDL particle contains 1,000 or more cholesterol molecules at its core. A monolayer of phospholipids surrounds the cholesterol core and is embedded with proteins called ApoB. These ApoB proteins are specifically recognized by the receptors in the cell's membrane. The receptors in the coated pit bind to the ApoB proteins on the LDL particle. The pit is reinforced by a lattice-like network of proteins called clathrin. Additional clathrin molecules then add to the lattice, which eventually pinches off a part of the membrane with the attached LDL particle inside. Inside the cell, the vesicle soon becomes uncoated as the clathrin molecules dissociate from the vesicle and from each other. Although not shown here, another vesicle then fuses with the LDL-containing vesicle, causing the pH inside to drop and forcing the receptors to dissociate from the LDL particle. After the receptors are released from the LDL particle, the vesicle fuses with a lysosome. A lysosome carries enzymes capable of digesting the LDL particle into amino acids and fatty acids. At the same time, the cholesterol is released into the cytoplasm. The cell either uses the cholesterol immediately or stores it for later. Meanwhile, the cell's LDL receptors are returned to the plasma membrane. Once in the plasma membrane, they can take in more LDL particles by receptor-mediated endocytosis.
Okay, so I hope this was clear. Okay, so this was just a representative using LDL particle. So LDL particle, as mentioned earlier, okay, in the beginning of this clip, okay, these cholesterol molecules are wrapped in the phospholipoprotein core. Okay, so that structure mimics the liposome or some solid lipid nanoparticle. Okay, and they bind with the LDL receptor, which are overexpressed onto the biological cell. Okay, and in that way they are internalized and they are processed. Okay, so this happens in case of all kinds of nanoparticle. Okay, so all kinds of nanoparticle, okay, they are capable of going or entering inside the cell by the same receptor mediated endocytosis. Okay,
In contrast
so
to
after that,
endocytosis,
degraded particles are
exocytosis
removed
is the
from the
process
cell by
which a variety of
exocytosis.
substances leave the cell. For example, newly made proteins destined for secretion are packaged into vesicles inside the cell and transported to the plasma membrane. The vesicle membrane and plasma membrane fuse together. The contents of the vesicle spill outside the cell, while the vesicle membrane is smoothly incorporated into the plasma membrane.
Okay, so in the second clip, you have seen how this degraded nanoparticle or degraded cholesterol, it is thrown outside the cell by the exocytosis. Okay, so cell doesn't keep anything inside. Okay, so whatever the material it engulfs, okay, it is degraded and it is then thrown outside the cell by exocytosis. Okay, so this is the routine process happens. Okay, so this endocytosis is the key of targeted drug delivery. Okay, so every nanoparticle or this type of nanostructure, it is capable of internalizing by the cell by the receptor mediated endocytosis. Okay, so different type of receptors are overexpressed or expressed onto the cell surface. Okay, some receptors are overexpressed in the case of particular disease condition that binds with the particular molecule. Okay, so what we need to do? Okay, we need to identify. Okay, this is the particular receptor overexpressed onto the target cell. Okay, so like folic acid receptors, these are overexpressed onto the tumor cell. Okay, so if we can conjugate the folic acid molecule with our nanoparticle, okay, we can ensure that they will selectively bind with the receptor present onto the tumor cell, and they will be internalized by the same process. Okay, so this ligand receptor interaction, okay, it is the key of targeted drug delivery. Okay, so again, by using this multifunctional approach like conjugating with some side directing ligand, we can achieve the targeting. But the major question comes how to bypass the RES uptake. Okay, as I mentioned earlier, this plain nanoparticle, okay, they will not go anywhere inside the body. They will simply go into the liver and spleen, that is reticuloendothelial system, where they will be degraded, or this they are engulfed by the macrophages. Okay, and they are degraded. Okay, so for this, what we can do the pegylation. Okay, here is the role of polyethylene glycol. Okay, so by pegylation, we can improve the hydrophilicity of the nanoparticle. Okay, so this if we make this nanoparticle in as a hydrophilic in nature. Okay, so this opsonin protein, okay, they cannot recognize the actual surface of the nanoparticle. Okay, so this pegylated nanoparticle, okay, they would be circulating in the blood for prolonged period of time. They would have a long half-life. Okay, and these are also called stealth particle. Okay, so they will have enough probability to go into the this tumor site. Okay, so it will bypass the RES uptake, and it will they will go into the tumor vasculature. Okay, so the marketed formulation, doxil formulation, that is pegylated liposome of doxorubicin, it is available in the market. Okay, so which is based on the same concept. Okay, pegylated nanoparticle, these are very popular nowadays. Okay, and if we can have this side directing ligand as well, so we can achieve the targeting. Okay, like they will selectively bind with the target cell, but onto the normal cell they do not have receptor. Okay, so they will not bind with the normal cell, so the toxicity or side effects to the normal cell can be avoided. Okay, so what I want to convey here, okay, if we have the plain nanoparticle, okay, they will show rapid clearance from the circulation. Okay, higher uptake by the RES organ, liver and spleen. They do not have any target specificity. Okay, so this plain nanoparticle, okay, these are basically useless system, except they are good for treating the liver associated disease. Okay, like if some liver disease, fatty liver is there, or some like liver cancer is there, or some other liver associated disease, leishmaniasis is there, then these are good system, but otherwise these are not a suitable system. Okay, to generate the target specificity, okay, we can conjugate with some side directing ligand. Okay, but again, okay, the major question comes that the RES uptake cannot be avoided. So this will show rapid clearance from the circulation and higher uptake by the liver and spleen or RES organ. To bypass the RES uptake, we can go for this stealth or long circulatory or pegylated carrier system. Okay, but again, this pegylated carrier system, okay, they do not have any target specificity. Okay, so to enjoy the maximum targeting efficiency, we should go for this multifunctional approach where both pegylation and ligand anchoring can be done simultaneously. Okay, so by pegylation, we can prevent the RES uptake, we can increase the circulation half-life, and by ligand anchoring, okay, we can achieve the targeting. Okay, so this last system, multifunctional system, these are most useful for the targeted drug delivery. So based on the same concept, okay, we developed this albumin, human serum albumin nanoparticle containing methotrexate, and they were conjugated with polyethylene glycol PEG and folic acid. So folic acid as a targeting ligand, and a PEG to make them long circulatory. Okay, so this is the AFM image of the nanoparticle. These are the scintigraphy images of the nanoparticle, okay, showing the biodistribution pattern. Okay, so here in one leg we implanted tumor. So with the free drug, you can see, okay, nothing is going inside the tumor, or very negligible amount is going into the tumor. But with this final formulation, significant amount was going inside the tumor. Okay, and if we just compare in the quantitative terms, okay, so tumor to muscle ratio, that is target to non-target ratio. So in the case of plain nanoparticle, it is 2.0. In the case of this targeted nanoparticle, it is 40. Okay, so 20 times higher retention at the tumor site. Okay, it is evident from this quantitative data. Okay, on the similar line, we prepared this liquid crystalline nanoparticle. Okay, so these are made of some special lipid. Okay, so they form self-assembled nanoparticle. Okay, this is the cubic shaped nanoparticle. Okay, so they self-assemble. Okay, so like in the case in the two form, hexagonal phase or cubic phase. So if they are self-assembled in the cubic shape, okay, then these structures are also called cubosome, otherwise they are called hexosomes. Okay, so three formulation: docetaxel to plain nanoparticle, pegylated and folic acid containing nanoparticles we are prepared. Okay, and we got good entrapment efficiency close to 80%. Okay, so that one, and the particle size was less than 200 nanometer. Okay, so when we checked the anti-cancer efficacy of this type, particle, okay, so we implanted tumor into the rat, okay, and we divided the tumor being a rat into the two groups. One is a small tumor and another one is the large tumor. Large tumor where the tumor size was greater than 1000 millimeter cube, and in the small, the tumor size was less than 1000 millimeter cube. So in the case of a large tumor bearing animal, okay, so this marketed formulation, that is Taxotere. Okay, so it shows approximately 20% reduction. Still 80% tumor was remaining. Okay, but in case of our formulation, it shows approximately 70 to 80% reduction. Only 20% tumor was remaining. And in the case of a small tumor bearing animal, okay, we got complete regression of the tumor just after 4 days of the treatment. Okay, but the marketed formulation Taxotere, okay, it gave approximately 50% reduction in the tumor volume. So our formulation was even more efficacious as compared to the marketed formulation. Okay, for this formulation we got the Indian patent. Okay, so this Taxotere, the marketed formulation of the docetaxel, it is a very toxic drug as well. Okay, toxic. The drug is not toxic. The main toxicity because the docetaxel, it is not soluble in the aqueous media. Okay, so this Taxotere contains, okay, this ethanol and twin-80 as a solvent. Okay, so twin-80, which is a very strong detergent or surfactant, and along with ethanol, both are not at all recommended for intravenous injection. Okay, but this formulation is approved by FDA because as an exceptional case, because it can increase the lifespan of the patient for few days or few months. Okay, but otherwise it causes severe toxicity. So we also evaluated the toxicity. Okay, but I'm not going to present that data because of the time limitation. Okay, so next case study I would like to present on the carbon nanotube for tumor targeting. So carbon nanotube, it is also a very attractive carrier nowadays. Okay, because of its some unique properties. Okay, like it is the unique needle-shaped geometry. Okay, so they have a high cell-penetrating power. So cellular internalization is very easy for carbon nanotube. Okay, and it shows a very high drug loading because of the pi-pi stacking interaction. Okay, so chemistry people can better understand what is this pi-pi stacking interaction, but it shows a very high drug loading. Okay, so as compared to the other nanoparticle. Okay, the major problem with carbon nanotube, it's they are not soluble in aqueous media. Okay, it is not soluble. If you just disperse the carbon nanotube in the aqueous media, it tend to settle immediately. Even if you try to disperse them with the help of some surfactant or some solubilizer, then also, okay, they tend to settle. The dispersion is not very much stable. Okay, you can also do sonication to disperse the carbon nanotube, but that approach is also not successful. Okay, but and another problem is they are very toxic. The toxicity is because of their insolubility, and they penetrate into the vital organ, and they just accumulate in the organ. So this is the problem. But this problem can be resolved by functionalization of the carbon nanotube. Okay, so for functionalization, different methods are proposed, but I simply use this oxidative functionalization. So these carbon nanotubes we are heated or boiled in the concentrated acids. Okay, that is the aqua regia. So upon this acid treatment, okay, so it generates the hydrophilic carboxylic acid group onto the surface because of the oxidation. Okay, so this hydrophilic groups, carboxylic acid group, they improve the hydrophilicity, and further they give opportunity to conjugate with the polyethylene glycol, okay, to achieve the biocompatibility and to achieve the further stealth characteristic and to make them target specific. Okay, so these pegylated or oxidized carbon nanotube, okay, they showed the enhanced dispersibility and improved biocompatibility. Okay, so initially when we started this type of experiment, functionalization, okay, so we did a lot of optimization, the functionalization for how much duration, because different reports says, okay, in some reports it was mentioned only for 2 hours, in some reports it was mentioned overnight oxidation, in some reports it was for the 12 years, 12 hours. Okay, so it was really confusing. Okay, and another reason, the carbon nanotubes can be procured from different sources. Okay, so initially we try to optimize this oxidation time. Okay, so we did several tests. This is just the qualitative test I'm showing. Okay, so this is the same image, scanning electron microscopic image of the carbon nanotube. Okay, after 2 hours of oxidation, aggregation was somewhat reduced, but still we can see a lot of aggregation in the side panel. This is after 6 hours of oxidation, so you can see this individual carbon nanotube, well dispersed carbon nanotube we can see. And after 8 hours, okay, the structural integrity of the carbon nanotube was compromised. Okay, so they tend to dissolve instead of dispersing, they tend to dissolve in the acidic medium. Okay, so we optimize 6 hours as good oxidation time. This is high resolution TAM image. So this is the multi-walled carbon nanotube. We can see different walls of the carbon nanotube as well, and we can also calculate the distance between the 2 walls. Okay, so the overall thickness of the carbon nanotube, okay, can be determined. Okay, then we loaded with the drug. Doxorubicin was loaded into the carbon nanotube. So different concentrations. So doxorubicin is a fluorescence drug molecule. So it shows some fluorescence spectra, okay, fluorescence intensity. But when the drug is encapsulated in the carbon nanotube, so the intensity of the fluorescence was reduced. Okay, that is known as fluorescence quenching. So the maximum fluorescence quenching or intensity was reduced at 2:1 carbon nanotube to drug ratio. It means this 33% of the drug can be loaded into the carbon nanotube, which is a very huge amount. If you see the label claim of the liposome, it contains approximately 3% of the doxorubicin, but here we could load 33% of the doxorubicin. Okay, so loading efficiency was tremendously improved with the help of this carbon nanotube. Okay, when we checked the in vivo efficacy, so you can see this doxorubicin loaded inside the carbon nanotube shows the minimum residual tumor burden as compared to the free drug or other formulation. Okay, we also evaluated toxicity. So liver toxicity was evaluated by measuring the liver function test. Okay, like AST level or ALT level, which is also known as GOT or GPT level and MDA level. In the case of toxicity, the level increase or decrease above or below the normal value, as we can see in the case of this pristine or non-functionalized carbon nanotube. But after functionalization, the values of these biochemical parameters was close to the healthy group. So we can see the toxicity was significantly reduced. Okay, then we went for the multifunctional system. Okay, so we choose this like a pegylation and a hyaluronic acid. So hyaluronic acid was used as a targeting ligand. So hyaluronic acid, it is a very good ligand for the CD44 receptor, which are overexpressed onto the some tumor. Okay, so this was the scheme, synthetic scheme we followed. So initially this plain or pristine carbon nanotube, we are oxidized. Okay, functionalized. So these are the oxidized carbon nanotube. This carboxylic acid group we are activated using standard thionyl chloride chemistry. Okay, then they were reacted with this bis amino PEG. So PEG on PG contains amino group on both sides. So this one amino group was reacted with this acid group, okay, to give this amide linkage, and these are the pegylated carbon nanotube. Okay, and this contains one free amine group as well, okay, which was conjugated with hyaluronic acid, carboxylic acid group of the hyaluronic acid. So these are the HA functionalized carbon nanotube, and doxorubicin was physically encapsulated, okay, and this gives the final formulation. Okay, we also did all the characterization studies in terms of FTIR, thermogravimetric analysis, X-ray photon spectroscopy, and NMR analysis, whether this conjugation was done or not. Okay, I'm not showing all these detail. I'm just showing the key findings. This is the cell uptake of this nanoparticle formulation. Okay, so these are the pegylated formulation, and these are the hyaluronic acid containing formulation. So hyaluronic acid containing formulation, you can see the degree of uptake was significantly high, because here you can see more number of cells. Of course, it was not like this plain carbon nanotube can also internalize by the cell by non-specific mechanism. Okay, we cannot stop. Okay, but here the degree of uptake was very high, and here we can also see if we pre-incubate the cell with a free hyaluronic acid to saturate the receptor, uptake was completely blocked. You can't see any cell here, no uptake. Okay, but in the case of plain carbon nanotube, okay, the uptake was not blocked after pre-treatment of the cell with free HA. Okay, so here it means they are following some different pathway. Okay, but here they are following this receptor mediated endocytic pathway. Okay, so how this pathway is important? Okay, this is explained in this slide. Okay, the target organelle of this doxorubicin action, it is nucleus. Okay, so what can happen? This drug can go inside the cell. Okay, but inside the cell, okay, there is nucleus, there is a lysosome, there is an endosome, it is simple cytoplasm also. Okay, so many organelles are there. Okay, so it is also important that drug should go, okay, with the nucleus after reaching the, after internalization by the cell. Okay, because if it does not reach to the nucleus, it will not act. Because, okay, doxorubicin, it inhibits the DNA synthesis. So ultimately it should reach up to the nucleus. So what we did, we stain the nucleus of the cell by DAPI. DAPI is a dye, okay, which if we incubate with the cell, it will go inside the cell and it will selectively stain the nucleus. So whatever this green structure we can see, these are the nucleus of the cell. Okay, and this red structure, these are the carbon nanotube. Okay, because carbon nanotube, okay, we labeled with some fluorescent dye Alexa 647. Okay, so this gives the red fluorescence. Okay, in the overlay image, what we can see, this red and the green, this red and blue fluorescence, it is perfectly overlapping. Okay, so what we can conclude here, okay, these carbon nanotubes and the nucleus, they are at the same location, or we can say the carbon nanotube or drug, it is at the nucleus side. Okay, the same analogy cannot be seen in the other image. Okay, so that's why here this HA functionality is very important. Okay, for targeted drug delivery. Otherwise, they can enter the cell by non-specific pathway, but it is not guaranteed that they will go with the nucleus or not, because we cannot see such overlapping here in the second image. Okay, and then we checked their capability to induce apoptosis or inducing cell death. So different tests we have conducted. Okay, like early apoptotic cell, late apoptotic cell, condensed nuclei, fragmented DNA. Okay, so different tests. Okay, series of tests we have conducted. Okay, so in the case of free doxorubicin, it gives some value, but in the case of doxorubicin loaded in the carbon nanotube, the values are always high. Okay, it means the doxorubicin loaded into the carbon nanotube, it induces higher degree of efficacy or apoptosis. Okay, then we conducted the in vivo studies as well. Okay, but I'm not showing the data here. Okay, so now move to the next topic, that is enhancing oral bioavailability via nanoparticle. Okay, so if I give you choice, okay, of course, no one want to take medicine. But if one has to take the medicine, whether you want to take the oral tablet or capsule, or you want to visit hospital and say, doctor, give me the intravenous injection, or I'm going to admit you, just infuse me the drug, intravenous infusion. So what would be the better option? Always oral tablet or oral formulation is best option in terms of patient compliance. Okay, but unfortunately, many drugs do not absorb following oral administration because of the bioavailability issues. Okay, so how this nanoparticle can help in improving the oral bioavailability, why the drugs are not absorbed from after oral administration. Okay, so I will just explain in this slide. Okay, so there are two types of cell present in our small intestine. One is called enterocyte and another is called M cell. So normal drug molecule, they are absorbed by this enterocytes via passive diffusion process or facilitated diffusion. Okay. So for that, the drug need to be present in the solution form. Okay, so solubility or dissolution of the drug is the first barrier. Okay, if the drug is not able to dissolve, if the drug is not able to solubilize in the gastrointestinal media, it will not be available for the absorption. Okay, so this is the first barrier. Second barrier is the permeability of the drug. Okay, not only the dissolution, but permeability is also important, because the drug need to cross this lipoidal barrier. Okay, so if the drug is not permeable, okay, it will not absorb. Okay, third thing can be if drug is not stable in the GIT. Okay, because some drugs, they are acid sensitive. Okay, like a penicillin antibiotic, they degrade at the acidic pH. Okay, so if the drug is unstable at the acidic pH, of course we can make the like entry-coated formulation. Okay, but some drugs, they are sensitive to the enzymatic degradation, like protein-based drug, insulin. Okay, insulin get degraded because of the enzymatic activity. Okay, proteinase enzyme trypsin, chymotrypsin, they degrade the insulin, or any vaccine, okay, which are protein-based formulation. So they are digested by the enzyme system. Okay, another barrier can be the P-glycoprotein efflux system, PG efflux system. Okay, so the PGP efflux, it is basically a efflux transporter, okay, which do not allow some toxin material to be absorbed inside the body. Okay, so what happens actually, many times we eat street food like pani puri, golgappa, samosa, okay, which is not at all hygienic. Street food, side mein road side दुकानें लगी रहती हैं, छोटे छोटे ठेले, okay, which are not at all hygienic. Okay, but we still eat. Okay, and we do not get any infection. Okay, so we should thankful to the PGP that PGP does not allow any toxin material, okay, contaminants present in the food, okay, to be absorbed into the systemic circulation. But unfortunately, many drugs, especially the anti-cancer drug, okay, they are also substrate for this PGP flux system. Sorry. So what happens, PGP just understand or recognize this is the toxic molecule. So if it will go inside the body, it will start killing of the cell. It would create harm to the body. PGP does not know if we have intentionally administered that drug. Okay, so this is the problem. Okay, so of course there are both good and bad facts about the PGP. Okay, but this is the fact. Okay, so many drugs, especially anti-cancer drug, they are substrate for this PGP flux system, so they are not absorbed into the systemic circulation. Okay, and another problem, that is the last problem, is the first pass metabolism. Everyone know. Okay, even if the drug is able to cross all these barriers, it may degrade in the liver, okay, before absorption, first pass transit. Okay, so all these problems can be resolved by nanoparticle. Okay, so when we encapsulate the drug inside the nanoparticle, okay, so the drug is not directly exposed to the enzymatic system, which cause degradation of the drug as well as acidic pH. Okay, so the pre-systemic degradation and metabolism of the drug can be prevented from these M cells. Okay, these nanoparticles are directly taken up by the M cells by process endocytosis, as I explained earlier. Okay, so there is no need for solubility, no need for permeability. Okay, because direct particle can be taken up by these M cells. Okay, so solubility, permeability related barriers are overcome. Okay, and the PGP also does not expressed onto this M cell, so this PGP is also bypassed, and from this M cells, okay, this these nanoparticle, they are absorbed into the lymphatic system first, and from lymph they enter into the main circulation. So it also bypass the first pass metabolism in the liver. Okay, I can explain further using this schematic. So when free drug is administered, okay, so either it will not absorb. If it is absorbed, and then it will first enter into the liver, where major fraction can be metabolized, which is known as first pass metabolism. But when these nanoparticles are administered, they follow the alternative path, okay, via lymphatic system. Okay, so they bypassing the first pass metabolism in the liver, and they are directly reaching into the systemic circulation via lymphatic system. Okay, so it bypasses the first pass metabolism. So in this way, it increases the bioavailability. Okay, and then besides this, there can be several other mechanisms, specific mechanisms for different type of nanoparticle. Okay, like for BCS class 2 drug, this or drug nanocrystal, they increase the dissolution velocity. Okay, for chitosan nanoparticle, chitosan is known as tight junction modulation. Okay, so it can increase the bioavailability, and so many other mechanisms are there, okay, which are specific. Okay, so what we did, we prepare a formulation for in this tamoxifen. Tamoxifen is a well-known drug for breast cancer treatment. Okay, so what happens after surgical removal of the breast? Okay, tamoxifen is provided, prescribed as a maintenance therapy. Okay, for 4 to 5 years. Okay, so, but tamoxifen, it is a BCS class 4 drug, poor solubility, poor permeability. It also undergoes first pass metabolism, extensive first pass metabolism. It is also substrate for P-glycoprotein efflux system. Okay, to because tamoxifen is prescribed as a daily therapy. Okay, like we need to take daily tamoxifen dose. So patient cannot go to hospital daily to take the tamoxifen injection. So that's why this tamoxifen citrate tablet, it is available in the market. So citrate tablet, citrate salt. So because citrate salt is relatively having higher solubility, so that's why it is prescribed. Okay, so but again, it can undergo precipitation in the acidic environment of the stomach. Okay, so it is having several limitations, but considering the practical consideration, key patient need to take this tamoxifen daily for 3 to 5 years, and it is not possible to call the patient every day at the hospital for 5 years. So tamoxifen citrate tablets, they are available in the market with reduced oral viability. The viability is just 10 to 20% only. Okay, so, and again, the problem with this tamoxifen, initially it is okay, but as I said, the poor bioavailability is because of the first pass metabolism. Every time when you are taking tamoxifen, it is passing through the liver, and liver need to do extra work to metabolize it during the first pass. So in the after chronic administration, after 1 year, it is start developing the hepatotoxicity, liver toxicity, because of the oxidative stress. Okay, so this is the problem with tamoxifen. Okay, so there can be a number of formulation approach which can be tried for improving the oral bioavailability of tamoxifen, but we thought of a very simple formulation, that is self-emulsifying drug delivery system. It is nothing but it is a mixture of oil, surfactant, and co-surfactant, in which the drug can be solubilized. Okay, like a vitamin E capsule, everybody might have seen. It can be filled in the soft gelatin capsule shell, and when we take this capsule orally, it forms nanoemulsion inside the body. Okay, without the need of any external agitation or homogenization, it forms nanoemulsion inside the body. That's why it is called self-emulsifying drug delivery system. Okay, so from industry point of view, it's a very simple technology. Okay, like a high level of industrial scalability, easy to manufacture. Okay, and it also increase the oral bioavailability, and similar type of formulation, they are available in the market for other drugs, not for tamoxifen, but for some other drug, the similar type of formulation, they are available. Okay, so what we thought, we will prepare the tamoxifen self-emulsifying system. Additionally, we have also incorporated quercetin in the formulation. So quercetin being antioxidant, okay, we incorporated. So what we thought, quercetin, okay, so it being antioxidant, it can reduce the hepatotoxicity of the tamoxifen because of the hepatotoxicity because of the oxidative stress. So antioxidant therapy can reduce the hepatotoxicity. And it is always like it is well known fact, if you combine the anti-cancer therapy with antioxidants, so it act as in a synergistic manner. Okay, so this was the reason we have included quercetin. So we prepared one particular formulation after a lot of optimization. Okay, so all the excipients were within the regulatory limits, like IIG limit. Okay, particle size was less than 200 nanometer. Okay, so when we checked the oral bioavailability, okay, so it increased the oral bioavailability by 8-fold, 800%, as compared to the tamoxifen. Okay, tamoxifen cited, which is available in the market. Okay, we also prepared one formulation without quercetin. So without quercetin, the bioavailability enhancement was approximately 4-fold. Okay, so fourfold. So we were surprised because we added quercetin with the purpose that antioxidant will reduce the hepatotoxicity, but we found this surprising result. It is also increasing the oral bioavailability. Okay, then we looked at the literature, we found the property of the quercetin, and we found quercetin inhibit the PGP. Okay, so this is the possible reason. Okay, quercetin being PGP inhibitor, it also contribute to increasing the oral bioavailability. Okay, so this was kind of very win-win situation for us, like sone pe suhaga ho gaya wo. Okay, when we check the anti-cancer efficacy of this formulation, okay, so it significantly reduced the tumor progression. Okay, so in the case of tamoxifen cited, the marketed formulation, it reduced the tumor growth up to approximately like a 60%. Okay, but in case of our this SNET's formulation, it reduced the tumor burden up to 80%. Okay, only 20% tumor was remaining. Okay, we also checked the mortality of the animal, survival of the animal. Okay, so initially we stopped the treatment after 1 month. After 30 days, we stopped giving any medicine, and we kept the animal and checked the survival of the animal. But in the case of other like marketed formulation, we can see death at different time interval. But with our this developed formulation, no death can be seen. Okay, during the treatment period. Okay, so this was the also a significant finding. It increased the survival of the animal. And lastly, we checked the hepatotoxicity of this formulation. Okay, so like different biochemical parameters, liver function test, AST level, ALT level, GSH level, TBARS level. So as I mentioned, in the case of toxicity, the level goes up or down as compared to these healthy values. Okay, so as we can see in the case of our formulation, the levels were comparable to the healthy groups. So we can say our formulation was relatively less toxic. For this formulation, we got the Indian patent and we have licensed this technology to VAV Life Sciences for further development and commercialization. So let's see what will happen. Okay, next case study I would like to present on this oral delivery of doxorubicin using this liquid crystalline nanoparticle. So doxorubicin, again, a very important anti-cancer drug, but only intravenous formulation is available, oral formulation is not available, and even intravenous formulation is very, very toxic. It causes severe cardiotoxicity. And the reason for this poor oral bioavailability of doxorubicin, because it is a substrate for P-glycoprotein efflux system. Okay, so PGP does not allow it to cross the intestinal barrier. So this is the major reason. Okay, so we developed this liquid crystalline nanoparticle of doxorubicin. Okay, so we prepared two different formulation. Okay, like glycerol monoleate based and phytantoil. So two lipid we have chosen, and we prepared the formulation. Okay, so we also checked the stability of this formulation in the simulated gastric and the simulated intestinal fluid. Okay, whether this formulation, this nanoparticle formulation, okay, they remain stable in the GIT or not. Okay, so they were stable in terms of particle size and in terms of entrapment efficiency. Okay, and we also checked their permeability. intestinal permeability in vitro using cell line, that is the CACO2 cell line. Okay, so you can see a significantly higher uptake can be seen inside the CACO2 cell, but a free drug, it was not able to internalize by the cell. Okay, so in the quantitative terms, the permeability was increased by 17-fold and 11-fold in the case of phytantoil and glycerine monoleate based formulation. Okay, we also checked their anti-cancer potential or uptake by the anti-cancer drugs. Okay, like MCF7, which is a breast cancer cell line. Okay, so intracellular trafficking was also checked because, as I mentioned earlier, for doxorubicin, the target organelle is nucleus. Okay, so we stained the nucleus and we found, okay, it is going with the nucleus. Okay, so approximately 75% co-localization with the nucleus can be seen. So these blue and this blue fluorescence is the DAPI stained nucleus, and this red fluorescence is because of the doxorubicin. Okay, and we also checked the possibility whether it is co-localizing with the mitochondria
बरं.
or it is
ठीक आहे, मी
co-localizing
बघू
with
उद्या
the
काय तो किती पेजेस
lysosome or
होते.
not. Okay,
झालं ना
so we
तुझं?
use
बरं,
these
ठीक आहे.
different
जायचं असेल तर जा
dyes.
मग. उद्या फक्त मला आणून दाखवाल ते सगळं. सेटिंग
Someone
करून
is
सगळी.
talking. Okay, so in this case, we did not find that a significant co-localization, just around 21% and 11% with the lysosomes.
Sir, please unmute yourself. अब सबको करना पड़ेगा.
Is that fine?
Yes, sir. Yes, sir.
Okay. Okay. Yeah, so we also checked the in vitro cytotoxicity by MTTSA, and we found the minimum IC50 value in the case of this doxorubicin-based formulation. So around 9.6 and 16-fold reduction in the IC50 value we can see here. Okay, and when we check the in vivo bioavailability or pharmacokinetic, so the bioavailability was increased by approximately 9-fold and 17-fold in the case of glycerine monoleate based formulation and this phytantoil based formulation. Okay, this was also a very remarkable improvement in the oral bioavailability of doxorubicin. Okay, next case study I would like to present on this insulin delivery, oral insulin delivery using the same liquid crystalline nanoparticle formulation. Okay, so we prepared this insulin loaded liquid crystalline formulation. We got approximately 250 nanometer as a particle size and approximately 40 or 45% entrapment efficiency. We also checked the stability in the simulated gastrointestinal fluid. So the formulation was relatively stable in terms of particle size and this entrapment efficiency. Okay, here we got this hexagonal shaped particle. Okay, we checked the stability of the insulin because insulin is a protein molecule, and during the preparation condition use stirring or homogenization, use of some organic solvent, okay, use of some surfactant as well. So it is possible that insulin get physically degraded, or it's a three-dimensional stability, tertiary structure get destabilized, secondary and tertiary structure, and they are destabilized. So that aspect was also checked using this electrophoresis and CD spectra sulfurous diachronis. So in the electrophoresis also we got single visible band at the same position. So we can say the insulin was chemically not degraded. Okay, and in the CD spectra we got superimposable CD spectra. Okay, so we confirm this there is no change in the secondary or tertiary structure of the insulin. So insulin remain biologically active also. Okay, and we checked the CACO2 cell permeability. So this is clearly visible. Okay, insulin labeled with FITC, that is a fluorescein molecule. So significantly higher uptake can be seen with the LCNP formulation. Okay, which is not there in the case of plain insulin. Okay, so significantly increased improvement in the permeability. And these are very interesting finding of the anti-diabetic activity. Okay, this is with the subcutaneous injection of the insulin, that is the standard one. So it lead to rapid reduction, rapid fall in the blood glucose level, but it is for the very short period of time. Within few hours, it restore to the normal value. But with our oral insulin formulation, okay, it causes the reduction in the blood glucose level for prolonged period of time. Okay, for more than 12 hours. Okay, it was found to be in the hypoglycemia. Okay, so subcutaneous insulin causes transient hypoglycemia up to 86%. Okay, so sometimes it causes severe hypoglycemic shock, okay, which is also not desirable, okay, because patient may die because of the hypoglycemic shock. So that's why it requires careful monitoring of the insulin dose. But in our case, oral insulin, okay, it reduced the blood glucose level up to 64%, and it was for up to 12 hours. Okay, so it is a well controlled hypoglycemia for prolonged period of time. Using some standard mathematical calculation, okay, we calculate the cumulative hypoglycemia. So for subcutaneous or injectable insulin, it was around 23%, but for our oral formulation, it was around 47%. So it was almost 2 times higher. Okay, the only thing for the oral insulin, okay, we took the dose of 50 international unit per kg, that was 10 times higher. Okay, but in the case of subcutaneous insulin, we took 5 unit per kg dose. Okay, so if we consider the dose, okay, if we consider the dose and we consider the bioavailability of this subcutaneous or injectable insulin as 100%, then we can say that our oral insulin only 20% bioavailability is there. Okay, but if we ignore the dose, okay, then we can say that our formulation was 2 times higher effective, or it is showing 2 times better bioavailability. Okay, so of course, lot of research need to be done to match the bioavailability at the same dose level. So that we are going on. So for this formulation also, we got the Indian patent, okay, in around few years back. Okay, another case study I would like to present on this oral mucosal immunization. Okay, this is another application in vaccine delivery using this nanoparticle. Okay, so as of now, okay, except for the oral polio vaccine, all other vaccines are administered through intramuscular or subcutaneous injection. Means all are the injectable vaccine except for the oral polio vaccine. So which is having several drawbacks, like these are expensive due to the need of like sterile manufacturing process and need of qualified medical personnel to for administration of the vaccine. This is the fact. All most of the participants, they would be having small kids in the family. Okay, so the vaccine cost is approximately 100 rupees. When you go to the some private clinic for getting the vaccine shot, so he will charge another 100 rupees. Okay, कि ये वैक्सीन की वो लगाने की फीस है और ये हमारी 100 रुपए की वैक्सीन की फीस है. Okay, so it is the cost is almost double because of the we require trained medical personnel and sterile manufacturing process for these syringes and other things. Okay, this is associated with a risk of a needle-borne infection like HIV or hepatitis if we use contaminated needles. Of course, this is not the problem nowadays, but earlier I have seen when I was kid. Okay, so this when this vaccination drive or vaccination camp used to be organized. Okay, so they used to come with like a single glass needle. Okay, and they used to mass vaccinate people of your entire family using the same needle. Okay, and sometimes they boil the needle, sometimes they do not boil the needle. Okay, so this was the problem. But of course, I'm not sure, but still this might be a problem in the rural area even. Okay, and of course pain, trauma, injection side reactions are there. Okay, so that lead to development of the needle phobia in the children. A very important fact about this injectable vaccine, they induce only systemic immunity, that is IgG type of antibodies, okay, which are present in the blood or serum. They fails to induce the local immunity, that is IgA type of antibodies, okay, which are present or which should be present at the common mucosal site, which are the majority of the pathogen enter inside the body. What I want to convey here, how do we get infection? Okay, we get infection either because of the contaminated food and water, that is the oral mucosal route, either because of the polluted air, okay, that is your polluted air, that is the respiratory route, nasal route, like your COVID infection is spread, okay, or because of the unsafe sex, okay, that is the genitourinary route. Okay, these are the three major sites, mucosal site, okay, oral mucosal, respiratory or nasal mucosa, and your lastly the genitourinary tract. Okay, so what happens in the case of conventional vaccine? Okay, these infectious pathogen, they enter inside the body by this mucosal route. I said oral mucosal or respiratory or the third route. Okay, and when they reach inside the blood, okay, inside the blood, because you have vaccinated, so you have antibodies, so you do not develop the disease. Okay, but what would be more advantageous? More advantageous would be if you can generate the antibodies at the mucosal surface itself. Okay, so that can neutralize the antigen immediately upon their exposure. Okay, these are called IgA, secretory IgA type of antibodies, which are present at the mucosal surface. Okay, but if you use this injectable vaccine, they are not able to generate this mucosal antibodies, IgA type of antibodies. Okay, so if you want to generate or if you want to develop this IgA type of antibodies, you need to give the vaccine through this oral route or respiratory route or nasal route or through any of the mucosal route. This is very important. Okay, but again, the same problem is there, like a vaccine, these are sensitive to the acidic pH. Okay, they are not permeable, they are degraded by the enzymatic system like insulin or any other small molecule we have just discussed. So it is very difficult to deliver them through oral route. So what we did, we prepared the chitosan nanoparticle. Okay, and we stabilized the chitosan nanoparticle. Okay, using some cross-linking, glutaraldehyde cross-linking we have done, because chitosan, it is soluble at the acidic pH. Okay, so if you prepare the chitosan nanoparticle and if you give through oral route, it will dissolve simply in the acidic pH. So we made cross-linked nanoparticle and we stabilized them with the help of glutaraldehyde. And further, to make them target specific, okay, so we manosylated the nanoparticle by glucomannan. Okay, so these four formulation we have prepared, and we use tetanus toxide as a model antigen to prepare this or to develop this formulation. Okay, so it was very easy to prepare the nanoparticle. Okay, and the particle size was approximately 150 or less than 200 nanometer. Entrapment efficiency was more than 90% in all the cases. Okay, so chitosan is a very good polymer to make the vaccine or protein nanoparticle, and it always shows more than 90% of the entrapment efficiency. These are different SAM and TEM images of the nanoparticle. Okay, we checked the stability of the antigen using electrophoresis as well as CD spectra, like we did in the case of insulin, and we did not found any degradation or change in the tertiary or secondary structure of the tetanus toxide or protein. Okay, we checked the antibody generation. So we gave the vaccine. This is the tetanus toxide. Okay, marketed vaccine which was given intramuscularly, injectable vaccine, and all these formulations. So this was our final formulation, that is the pink color. Okay, you can see. Okay, so our oral formulation. Okay, so obviously this marketed formulation or injectable vaccine, it induced the highest antibody titer in the blood. Okay, but our oral vaccine was also inducing the significantly higher antibody titer. Okay, although it was less as compared to the marketed one or injectable vaccine, but it was comparable. It was also very high, or we can say it is sufficient. Okay, and of course, initially at 0 day we give the first dose. After 21 days or 3 weeks, we give the booster dose. Okay. This is very important finding, secretory IgA titer or antibodies, this which is responsible mucosal immunity. Okay, so in the salivary fluid, in the intestinal secretion, in the fecal content, Okay, where this intramuscular or injectable vaccine failed to induce this antibody, our orally administered vaccine, they significantly induced, okay, this IgA type of antibodies, which is more desirable. Okay, so our oral formulation did not only induce the systemic immunity, but it also induced the local immunity, which is more advantageous. Okay, which we want to hypothesis want to prove. Okay, the last case study I looked like present on topical delivery. So Kalonji oil based nanoemulsion containing tacrolimus for synergistic anti-psoriatic activity. Okay, so psoriasis is again a well-known problem. It is a dermatological problem. Okay, so tacrolimus, it is a well-known drug. Okay, it is an anti-inflammatory drug. Okay, which it is the mechanism is clear. Okay, it is an anti-proliferative and anti-inflammatory drug, so it is recommended for psoriatic lesion. Okay, so we need to prepare some formulation. So what we did, we prepared the nanomulsion using this natural oil, Kalonji oil. Kalonji seeds, everybody might have seen. So the active component is thymoquinone. Okay, so thymoquinone, it is also having a anti-apoptotic effect. Okay, it is an antioxidant. It is having some, it induce some pro-inflammatory cytokines. So it is a anti-inflammatory, anti-apoptotic agent. Okay, so it can help in giving the synergistic activity. Okay, so that's why we have selected this Kalonji oil. And fortunately, our drug was also showing a very good solubility in Kalonji oil. We of course tried several oils. Okay, but best solubility was observed in Kalonji oil. Okay, so we could able to make this nanoemulsion formulation. We also applied the quality by design approach to optimize this nanoemulsion formulation. Okay, so when we checked the in vitro skin permeability, so we got the best permeability using this our nanoemulsion, and nanoemulsion was also incorporated into the some topical gel. So both nanoemulsion and nanoemulsion gel, they showed good skin permeability as compared to this free drug. Free drug, when it was incorporated into gel, it was simply sitting onto the surface of the skin. Okay, but here this nanoemulsion, they are and deeply penetrated inside the skin. Okay, so we checked the efficacy in the psoriasis model as well. Okay, so we induce psoriasis, okay, using this imiquimod model. Imiquimod cream was applied, so it generates psoriasis after in 1 week. Okay, so different formulations, like this is a healthy control and marketed formulation, that is the tacrolimus gel was also used, and this is our final formulation, H formulation. So it shows a significantly or very good efficacy in the psoriasis model. And in collaboration with one hospital, we also tested this formulation in some human subject, and it shows excellent result. Okay, this is initially, this is after 30 days, this is also after 60 days, and this is after 3 months. Okay, so it is almost clear. Okay, this is the photograph of leg, this is of the hand. So in the hand also, so it is almost clear. Okay, so for this formulation also we have filed the Indian patent application, and let's see what will happen in future. Okay, so I would stop here. These are the key research area of my lab. Okay, multifunctional nanocarriers for targeted drug delivery. Okay, gene delivery via non-viral vectors, non-invasive vaccine delivery, oral bioavailability enhancement, topical and transdermal drug delivery, for delivery of anti-cancer drugs for synergistic efficacy, and lastly, the lipidic products for effective drug delivery. So we did a lot of work on amphotericin B delivery and cisplatin delivery using lipidic product. Okay, and this is the hypothetical design of ideal multifunctional nanocarrier system, which is capable of simultaneously diagnosing, therapeutic, delivering the drug molecule and monitoring the treatment. So more than one drug molecule it can deliver. It is a ideal multifunctional system for theranostic application. So if you have understood anything out of my presentation, you can think how can you design for your application this type of system. Okay, these are the 2 books published by me. Okay, one on targeted drug delivery by Springer, and another one is elsewhere, that is on cancer nanotechnology. So these are very good reference book. They should be present in all the code libraries. And lastly, these are the prestigious moment of my life when I received this Young Scientist Award and this AAPS Fellow and INSA Young Scientist Award. And thank you very much for your kind attention. And if you have any question, okay, we can take some questions.
Now session is open for question and answer. The session is open for question and answer.
So probably you need to unmute the participants.
They can unmute themselves, sir.
Okay, okay.
Yes, if anyone want to ask the question, ask right now. Yes, sir, one question in chat box.
Okay.
Which nano formulation will you suggest for phytoactives?
For phytoconstituents. Okay, so it depends on the again the solubility or physiochemical properties of the your phytoconstituents. Okay, so liposomes are also good, but again the loading in the liposome it is sometimes difficult, so we can go for some lipid-based nanoparticle.
Sir, can I ask one question? Hello?
And
Hello.
another challenge in the phytoconstituents, okay, you should have the purified molecule. Okay, purified molecule, because sometimes this crude extract, okay, crude extract, they contain multiple ingredients. Okay, so this is always very tricky, which means it is difficult. How will you determine the encapsulation efficiency? As you mentioned, it is a BCS class 2 drug, then definitely your solid lipid nanoparticle or your this liposome would be a best formulation. And again, what would be your route of administration? If it is for the topical application, then nanoemulsion based formulation is the best. Okay, if it is for the injection, then a liposome or nanoparticle, liposome is the best. But if you are preparing for the oral administration, then you can go for this either self-emulsifying drug delivery system or some solid lipid nanoparticle or this nanostructured lipidic carrier NLC based system.
Yes, yes, Bodhankar madam, you can ask the question.
Sir, can you hear me? Hello? Can you hear me, sir?
Yes, Jain
Yes.
sir. Madam is asking the question.
Namaste,
Yes,
sir.
yes.
Namaste, sir. It was a nice talk, really. I want to ask you one question that you have prepared the self-emulsifying drug delivery system, isn't it?
Yes.
With the tamoxifen and quercetin. And
Yes.
I want to ask that is there any interaction between tamoxifen, because it is a class 4 drug and the bioavailability problems are already there in the class 4 drugs. And if we give in the combination, then is it producing any further, is it giving any further problem in the release of the drug? That is my one question. And in self-emulsifying drug delivery system, we use surfactant and co-surfactant in higher proportion, isn't it? So whether it is means it is having any effect on the efficacy of the drug.
Yeah. So for the first question, the first study we did before combining the two ingredients, we checked all the compatibility testing. okay, as per the ICH or means regulatory norms, and regarding the high concentration of the surfactant or co-surfactant. So I clearly mentioned, okay, all the excipients, okay, whatever the concentration was we have used, it was within the IIG limit, permissible limits for daily administration. Inactive ingredient guide is their IIG limit.
Mhm.
Yeah,
Okay, thank you, sir.
yeah. So that's why we were able to transfer this technology to one of the industry. Yeah, otherwise no one tested. Yeah.
Yes, so it is very difficult. We get the patents, but
Yes.
transferring the technology to
Yes.
industry is a difficult task,
Yes.
and I would be
Yes.
congratulating you for that. Thank you.
Yeah.
This feedback link is available in chat section. Participant feedback link is available in chat section. Now I thank Dr. Sanyog Jain sir on behalf of IPER for an enlightening presentation. I would like to express my sincere thanks to Dr. Sanyog Jain sir for giving an excellent coverage for nanocarriers for targeting and treatment elements. Now, we are all inspired by your great work, sir. Thank you so much for sharing your experiences of the research with us. Thank you, sir.
Yeah. Thank you.
Thank you, sir.
Once again, everyone. Yeah.
Thank you,
Thank you,
sir.
sir. Thank you.
Now, with this, I declare that this session is closed and I request to join the next session tomorrow sharp at 10 a.m. 10:30 a.m. 10:30 a.m. Please fill the feedback link available in chat section. Thank you, sir.
Yeah. Thank you. Thank you. Okay. Once again, thank you, everyone, and bye bye.